Ma'am, please start with the introduction. Ma'am, please start with the introduction.
Thank you, Mr. Majid. Very good morning to all the dignitaries and participants. Today, it is my pleasure to invite Dr. Gorakhmal, one of the most eminent scientists of Indian Veterinary Research Institute, to this training program. Sir is currently working as Principal Scientist and Station in Charge, IVRA Regional Station, Parampur. Let's welcome Sir. I'll start with a brief introduction about Sir. Sir has expertise in animal biochemistry with special emphasis on recognition of the milk of hill cattle and Gaddi goats of northwestern Himalayan region as source of potential nutraceuticals. Chemistry, biochemistry and proteomics of Gaddi goats and hill cattle milk, and enhancing keeping quality and nutritional value of native forages by ensiling and added phytochemicals. Sir has published more than 100 research and review articles in national and international peer-reviewed journals. Sir is life member of assorted and prestigious scientific bodies. With this very brief introduction, let us welcome Sir once again to deliver his lecture on the topic Bioactive Potential of Himachali Pahari Cattle and Gaddi Goat Milk. Welcome you, Sir. Please.
Thank you, Dr. Devi Gopinath. I would like to extend my thanks to coordinators, especially Mr. Majid Nisar and organizer of 21 days program for giving me opportunity to deliver lecture on the topic bioactive potential of Himachali Pahari cattle and Gaddi goat milk. So brief introduction is that the local cows which are earlier nondescript and these were not identified as specific breed. But in 2020, ICR Institute National Bureau of Animal Genetic Research NBAGR Karnal has officially recognized this Himachali cattle as a Himachali Pahari cow, and this is now registered as a breed, and this is distributed in 7 districts of Himachal Pradesh, and those districts are Chamba, Mandi, Kullu, Kangra, Sirmaur, Kinnaur and Lahaul Spiti. And this breed is known as for distinct features. It has unique features and qualities. However, milk production is low, and that is around 1 liter per day. So that is disadvantage. Otherwise, now Gaddi goat is reared in different localities of Himachal Pradesh, and it is also known as white Himalayan goat. And this is predominant goat breed of high altitude western temperate Himalayas, with its true home tract in hills of Himachal Pradesh. But its distribution also extends to Jammu and Kashmir and Uttarakhand. And the breed is amply described in terms of its geographical distribution, morphological characteristics, production attributes, and cytogenetics profiles. However, there is no information about milk characteristics of Gaddi goat. So, with this brief introduction, importance of milk, I am going to tell that milk has different biological activities. Those are antioxidant, antimicrobial, immunomodulatory activities, and also milk recently the main focus is on bioactive proteins, and bioactive proteins are a source of antioxidants, antimicroorganism activities, and Gaddi goat is also thought that it has low allergenic potential because it has proteins which are the protein that is casein alpha S1 casein. Its characteristic is different from cow milk, and therefore it has low allergenic potential. And it is found that milk with a higher antioxidant capacity have a higher oxidative stability and will provide protection who will consume this milk who will consume this milk, and oxidative stress will be reduced by consumption of milk in many acute and chronic diseases. So this is a major bioactive functional compounds present in milk. Those are mainly these are proteins. Mainly these casein and whey proteins are present, and those are major proteins. And when these are digested, then they will derived into peptides. Peptides are small amino acid sequences. Those are 3 to 20 amino acids in length, and those have this bioactivity. Another is immunoglobulins. Those are present in milk, and then growth factors, cytokines, all are present in milk. And then lactose and carbohydrates are also lipids, fats, minerals, vitamins. Lactoferrin, those are protective proteins, protective in nature. Those will provide protection to various diseases to human beings and other who will consume milk. And enzymes also present. Enzymes will provide the increase the shelf life and protective properties to milk. So I am going to discuss my talk with following subtopics. First, I am going to discuss about milk composition of these indigenous cattle and Pahari, this Gaddi goat, then antioxidant activity of milk, and then vitamin contents, then composition in terms of fatty acids, then effect of thermal processing on antioxidant activities, and then again heat effect on this protein profiles. Then this is a bit specific on this peptides activities present in milk, uh, and then effect of digestion on protein and peptide profiles, then antimicrobial activities, and lastly I will tell briefly about anti-hypertensive activities of milk of indigenous cattle, Pahari cattle and Gaddi goat. So first thing, milk composition, that is very important to study milk composition. So milk composition will tell us about idea that what are the constituents present in this milk as a total. And milk is a mixture of several substances. Those are lactose, protein, lipids, amino acids, urea, and many other things, creatinine, etc., are present in milk. And its composition also depends on many factors such as breeding programs, feeding schemes, and climatic conditions, and so many factors are important to determine the composition of milk. And bovine milk is so far has been studied so much as compared to Gaddi goats, and it has been mm, seen that composition of milk depends upon species and breeds of animals, just I was telling. And also it depends upon there is a variation in morning milk to evening milk, and if we will going to take milking in noon, then again it may be variation will be there in the composition. So ultimately composition depends upon many factors, but it is a gross composition, and it will give a fair idea about what is present in different types of milk. So first, if we will discuss about pH, then pH is around 6 to 6.71 and 6.83 in local gourds, and acidity. So pH and acidity give us idea about the shelf life of milk and how this can be extended. So then fat, total solid protein, lactose, these are important constituents of milk composition and important nutrients. And here I would like to say that in Gaddi goat, this fat milk, this fat percentage is higher as compared to other types of milk, and also protein that is low as compared to milk of indigenous cattle, Jersey crossbred, and local goats. So this was a brief about this composition. And it is also observed that this goat milk is preferred for infants as it has been observed that it has overall benefits in terms of health and immunity. And while doing this work, we have observed that people of Chamba and Kinnaur district of Himachal have been treating diabetes with a concoction made of Gaddi goat milk. And in our dialogue with the local people in Palampur and Simla, we have observed that Gaddi goat milk is also given to people that are infected with dengue. So, however, scientific evidence towards these claims are lacking, but there is a need for this careful scientific investigations. So here I am going to tell about that there is variation in milk composition early, mid and late lactation stages, and we can see that there is difference in the pH of milk in early, mid and late lactation, and this variation may be noted in all types of milk in indigenous cattle, Jersey crossbred cattle, Gaddi goat's milk and local goat's milk. Then SDT I was also going to tell about this again composition. However, these are variation are non-significant in early, mid and late lactation stages. Then next is fat percent, and here we can see that fat percent in late lactation was found to be highest in indigenous cattle, and here in Jersey crossbred cattle also it is highest, and in Gaddi goat that is also highest in here in local goat that in late lactation fat is highest. Then total solids, those are again in late lactation, those were higher, but again these were differences were non-significant. Then protein composition we have studied in early, mid and late lactation, and it was only significantly difference in late lactation in indigenous cattle, and there were no difference found in the other milk types. However, in Jersey crossbred it was higher as compared to early and mid and early lactation. Then lactose we have studied again. We can see that these lactose contents are variable, and no definite trend has been observed in these different types of lactation period. Then next is antioxidant activity. I am going to tell a bit about antioxidant activity. Antioxidant activity, when an excess of free radical is found in the body, they can overwhelm protective enzymes like superoxide dismutase, catalase, and peroxidase, and cause destructive and lethal cellular effects by oxidizing membrane lipids, cellular proteins, DNA, and enzymes, thus shutting down cellular respiration and cause oxidative stress. Oxidative stress plays a significant role in many age-specific diseases. Many studies have shown increased oxidative damage to all major classes of biomolecules, including these fat, protein, lipids, and in brain of Alzheimer patients, it has been found that these are oxidative damage is found. So here main emphasis is that what are antioxidant compounds activities, whether milk has antioxidant activities or they indirectly they can they have present those antioxidant compounds. So antioxidant compounds are considered to be essential in diets or medical therapies for delaying aging and biological tissue deterioration. And these days search is going on for antioxidants in biomedical sciences and as well as in scientific research. So antioxidant activities we have studied in different types of milk. Here I am going to tell that we have studied antioxidant activity in whole milk, and these are the fractions of milk, whey and casein. So actually in milk has two major protein fractions, whey and casein. So we have separated milk to whey and casein, and then we studied this antioxidant activities. So antioxidant activities, there are so many methods to study the antioxidant activities. One of them is FRAP, and here we have studied antioxidant activity with the help of this method. And here if we will see the overall data, we can see that overall antioxidant activities higher in casein fraction in all milk types as compared to whey fraction. So we can say that casein is the main constituents or contributor for antioxidant activities in milk, or we can also say that casein is an important contributor for overall antioxidant activity in milk in all milk types. So this again we have studied on the basis of different lactation phases, and we can see that variability we can see in early, mid, and late lactation stages antioxidant activity. And here this is the Bay fraction. Again, antioxidant activity is variable. Then this is Cajun fraction. So we can see that antioxidant activity is present, but uh, trend is not definite. So then there is another method to study the antioxidant activity, that is AVTS, and again it is a radical scavenging activity method. And here we can again see that antioxidant Cajun fraction is important contributor for this antioxidant activity. If we will compare these fraction protein fractions, then it is quite high, and as compared to this Bay fractions, and it is all found in all milk types. Then again, we have studied on the basis of the early, middle, and late lactation period, and this study was carried out in whole milk, and we can see that these are variable results are we have observed. Then this is the Bay fraction, and again we can see that results are variable, although antioxidant activity is present. So Cajun fraction, again results are variable in early, mid, and late lactation. Then there is another method, that is again it is a radical scavenging methods, that is Dpph method, and we have studied this by this method. Actually, to study antioxidant activity with various method, actually no method is specific, and no method will give a total antioxidant activity. So we have to get the fair idea about antioxidant activity. So we have studied three different methods, but all methods are showing that Cajun fraction has a higher antioxidant activity as compared to other this Bay fraction. So here we have studied this antioxidant activities in different lactation stages, and again this antioxidant DPPH radical scavenging activity is present in all milk types. However, trend is not specifically we can say, but significance is there. So then in Bay fraction, we can see that significant differences are there in early, mid, and late lactation. So here in Cajun, we have again studied an early, mid, late lactation, and we can see that the antioxidant activity is present in all lactation stages. So this is a qualitative method to detect antioxidant activity. So we can say that these are yellow spots showing that antioxidant activity is present. Here we were discussing about quantitative methods, but this is qualitative method. Yellow spots show that antioxidant activity is present in milk or control. So this was the control. Here we can see that yeah, these are bright spot. These are control, and that is ascorbic acid we have used here, and we can see that antioxidant activity is present. So these spots are after 30 minutes of spraying this DPPH on TLC plates, and this lane showing that antioxidant activity in overnight samples after 18 hours. So here we can see that these spots has been become brighter as compared to 30 minutes. So we can say that there are antioxidant present in milk. Those are slow reactant, and the nature of antioxidant compounds in milk are variable. And this antioxidant activity again we have seen in Gaddi goat and this Gaddi goat. And here this was in local indigenous cattle milk and Jersey crossbred. And here Gaddi goat and local goat. So we can see that spores are there, but those are also again shape and all those color may be different. So we have to search that what exactly these antioxidant compounds are present in milk, and that is an important topic we have to again investigate. Then this was skim milk. We have used skim milk where fat was removed, and just to see that still antioxidant activities due to this compound, those are present in fat or also in skim milk. So we can see that in skim milk also this antioxidant activity is retained. So next is vitamins. I am going to specifically discuss vitamin contents. Those are just related to antioxidant activities, not all vitamins. So here we have studied only vitamin C, vitamin A, and E. These are vitamins also involved in antioxidant activities. So these vitamins, vitamin E and C, and other carotenes and enzymatic systems such as superoxide dismutase, catalase, glutathione peroxide, these are present along with vitamins, and these provide antioxidant activities. So here we can see that in Gaddi goat vitamin C, that is fairly low as compared to other types of milk, and vitamin A, that is comparable, and vitamin E also, it is comparable with a bit higher in case of local goat. We can say that this is not a definite trend, but here in vitamin C we can say that it is fairly low in Gaddi goat milk. Then further vitamin C, as we observed that it is lowest in Gaddi goat. So we studied it in bit more detail in different lactation stages, and we observed that it remains lowest throughout the lactation period, and it is low as in early, mid, as well as late. In late, it is almost equal to indigenous cattle, but still we can say that it is not. It is to a lower side. So overall, I was told that I was telling that it is 0.88 milligram per deciliter. Then next is fatty acid composition. So fatty acid, these fat present in milk is most complex of natural fats, and more than 400 individual fatty acids have been identified in milk, and almost 98% of fat in bovine milk are made up of triacylglycerides, and the remaining approximate 2% is made up of diacylglycerides, cholesterol, phospholipids, and free fatty acids. The fatty acids in milk are derived from many sources, and just I was telling that it depends upon this composition of fatty acids, depends upon many factors, and important are breed and selection, lactation stage, I was discussing the parameters, then fermentation, then what type of feed we are offering, and season also play important factor, and then regional effects are also important. These are the major contributor for this fatty acid composition of milk. So here we can see that fatty acid composition we have studied in different milk types, and we can see that this saturated fatty acid, that is highest in local goat, and then in indigenous cattle. Saturated fatty acid is also present in Jersey, crossbred, and Gaddi goat. So in all this saturated fatty acid, that is not good for health, present in all milk types. However, unsaturated fatty acid, that is present in local goat and this indigenous cattle. That is again 1.72% and 1.15% in indigenous cattle. Otherwise, we could not be able to detect here in Jersey, crossbred, and Gaddi goat this PUFA. So this is a detailed composition. Individual fatty acids we have studied, and insaturated fatty acids, Caprylic acid is major contributor for this is for saturated fatty acid, and that is 15.74, and that is mainly present in indigenous cattle. And capric acid, that is highest amount is present in indigenous goat. And as I was telling, that this composition is variable, depend upon many factors. And then oleic acid, that is mainly present in oils, and that is so here it is present in Jersey crossbred, highest amount, and then Gaddi goat. And then it is fairly this is present in oleic acid is good for health also. So means this milk, these milk of indigenous cattle, Jersey crossbred and Gaddi goat, we can say that these are also source of this monounsaturated fatty acids. And then polyunsaturated fatty acids, again this we could not detect in detect and we got only in this indigenous cattle. So these are some chromatograms we are showing that we, with the help of chromatogram, this analysis was done with the help of chromatography, and we can see that these are the peaks, and with these peaks we can identify individual fatty acids present. So these chromatograms are entirely different. This is indigenous cattle chromatogram, and this is Jersey crossbred cattle chromatogram, and these are different to each other. And again chromatograms of local and Gaddi goat, again these are variable. We can say there's peak palmitic acid is present here, but here it is present on the other side. So then we have studied the effect of thermal processing. We have studied the effect of thermal processing on fatty acid composition of milk, and we have found that when milk was boiled, then this elatic acid, a trans fatty acids are not good for health, and it is not advisable to consume this trans fatty acid on regular basis. So here I would like to emphasize that when milk was boiled and this fatty acid composition was studied, then Jersey crossbred cattle milk has found to be highest this elatic acid. So we can say that Jersey crossbred cattle milk after boiling is showing this trans fatty acids, and least amount is present in this our Pahari cattle. And then Gaddi goat has in between. And moreover, when raw milk, we have not detected this elatic acid. I was showing earlier slides that it was not detected in raw milk of indigenous cattle, Jersey crossbred, Gaddi goat, and local goat. So we can say that if we have to boil the milk, then trans fatty acid is present, and those are not advisable for consumption. Then this is a composition again in boiled milk. So we can see that this is composition variable between different milk types. Here what I was telling about elatic acid, that is trans fatty acid, that is present in highest amount in Jersey crossbred, followed by Gaddi goat, and lowest amount here in indigenous cattle. It could not be detected in local goat. Actually the number of samples are limited, so we can, it is not definite. We have to study more and more samples, so then we can have a definite conclusion. Then next is I was telling about antioxidant activity. Usually we consume milk and other products are made after this pasteurization or boiling of milk. So we have studied this antioxidant activity after heating, or different types of various types of treatments were given to milk, and we studied the antioxidant activities after that. So here we have studied this total phenol, and we can say total phenol, again it is give some idea about the antioxidant activity. And we can see that in raw milk, this raw milk has this total phenol contents are somewhat lesser as compared to boiled milk. So boiling may lead to increase these antioxidant total phenol contents and in whole milk. And then this is a comparison of Gaddi goat and local goat. So here we can say here it is again here trend is reverse, and here we have noticed down that there is no increase in these total phenol contents after boiling. So this is earlier I was discussing that this is another method for this determining antioxidant activity, that is FRAB, and boiling led to increase in this antioxidant this activity in boiled milk in indigenous cattle as well as Jersey crossbred cattle milk. And then here but here we could not get any specific this increase. Moreover, it has been found to decrease. Then another method, Dpph, no definite trend we have observed here. Then in Gaddi goat and local goat, again it was not observed, trend was not observed. Then I was discussing that this is a qualitative method, and we have found that in boiling and all boiling raw and pasteurized milk, whether antioxidant activity is present or not. So we can see that these spots are present, yellow spots are present, and these are showing the antioxidant activity that is not destroyed even in boiling as well as pasteurization. So antioxidant activity in milk is retained after this boiling as well as pasteurization, and this was proved by this qualitative as well as quantitative method. Then another important thing is the proteins. Those are important parameter, nutritive parameter, as well as protein are these days main emphasis is given on the quality protein and bioactive proteins and bioactive peptides. So it is important to see that if we are heating the milk, and is there any effect on milk on proteins are destroyed or not, or if any conformational changes are happening during all those things. So we have studied this and it was found that usually we have observed that this cage infection, I was telling that cage infection, this heat treatment effect has not been observed. And in case of bay fraction of protein, those high molecular weight proteins, those are lactoferrin and bovine serum albumin, those were disappeared under boiling condition. And these are important observation, because earlier I was telling that casein are important for important contributor for antioxidant activity, and here also these are not destroyed during heat treatment. So this is again additional benefit. So here we can see that this is the whole milk. We have studied the profile. So in whole milk, this is a whole milk profile, and this is a pasteurized milk, and we can see that here this protein, this is a lactoferrin present at here. Those are disappeared in way after pasteurization, and otherwise these proteins, major proteins, these are casein proteins, those remain present throughout even after pasteurization as well as in boiling milk. And this was a milk we have taken from indigenous cattle, and then this was from Jersey crossbred cattle. Again we have this is raw milk, and we can see that this profile remains same, but here in lane 8, that is again boiled milk. So in boiled milk, this again I was discussing that these bands present here will disappear in boiling step. So lactoferrin is a protective protein, and it is a main protein, and these days lactoferrin is given and supplemented also, and we have seen in literature that from lactoferrin many bioactive peptides are commercially available, and they have got antimicrobial activity. One of them is lactoferricin, and that is available commercially, and it has been used as good as antibiotic. uh, so here, but here we can see that it has been not observed in boiled milk, although it is present in these pasteurized milk. Then another slide is this is local goat milk. Again, we can see that in local goat these proteins are present, but again this protein has been disappeared in boiled milk. So this is a Gaddi goat. Again, we can see similar trend here. This protein has been displayed. So the basic purpose to show these slides is that we can see that boiling has effect, mainly affect this wave fraction, and wave fraction is also important because branched amino acids, those are required for this for building of muscle. Those amino acids are present in a wave, but after boiling those may be destroyed. So but casein remain intact. So here we have tried to differentiate between the specifically between the milk of different types. So here we have used again milk of Gaddi goat and this indigenous cattle and local goat, and we can see that there is specific difference between these two milk, and these are this protein that 85, this protein, this band is present in a local goat and also present in this Gaddi goat. No, this crossbred goat, and these are some bands. Those are peculiar to these are bands which are these are peculiar. So here we have tried to identify, characterize those bands, those proteins. What are those proteins? So this was done by this Malditov. That is again a very costly equipment, and we have tried to done this work with the help of CSIR IHBT, that is Institute of Himalayan Bioresources Technology, and they provide extended help to us to identify those peptides with the help of this equipment. Actually, antioxidant peptide present in food chain play a major role in immune system by preventing formation of free radicals or by scavenging free radicals and active oxygen species. So earlier I was telling that antioxidant from natural sources are major are more superior to those produced chemically, because synthetic antioxidants such as uh, uh, synthetic antioxidants have been reported to be carcinogenic, cancerous effect. So these days scientists are emphasizing to identify natural antioxidants. Those can be isolated or purified from natural sources, such as milk is one of them. Then there may be another sources from plant, herbs, and other things. So these days emphasis is going on those resources those are locally available. So we have tried to identify those peptides present in milk. So here this is again different steps. Earlier I was telling that these are the bands present in milk. So we have tried to characterize these bands with the help of this method. So these are peaks. We can see that these are peaks, and these are showing that different peptides are present in different types of milk. So here these are the peptides. We can see that these are the peptides present in milk. Those bands which are unique in different different types of milk, we can see that these are peptides. Now peptides, what are peptides? Those are short sequences of amino acids present in milk. If protein native form, these peptides are not released, and when these are processed by with the help of enzymes, these peptides we can see, and these peptides are short sequences. So short sequences of peptides always revealed antioxidant or some health promoting effects. So these are the advantage of these short peptides, because they provide some health benefits. So if anyone have to study these peptides, they have to study those antioxidant, antimicrobial, anti hypertension, and so many activities to prove that these have some therapeutic activity, and those provide some beneficial effects, health beneficial or nutraceutical benefits to human beings. So these are the peptides. So next, we have studied this in vitro digestion, and in vitro digestion again, that what is in vitro digestion? We consume food, and in food proteins are present. So those proteins are hydrolyzed in gastrointestinal digestion, and before being transferred as small peptides to intestine enterocytes. So they are finally released into interstitial space and absorbed, finally absorbed. So milk proteins are a source of biological active peptides, and I was showing that those peptides earlier I was showing those were not digested, but here we have digested these proteins, and we try to search out that if new proteins are present, or new protein, these proteins are proteolized, and we get some new peptides or not. So we have tried this in vitro digestion. So how these bioactive peptides can be produced from major milk proteins? So this is just a brief. We can see that earlier I have discussed that caseins and whey proteins, these are the important main constituents of proteins present in milk, and these caseins are further subdivided and identified. We have seen these profiles in SDS space: alpha S1 casein, beta casein, kappa casein. These are the major proteins of caseins, and the whey proteins are further distinguished in as alpha albumin, beta lactoglobulin, lactoferrin, and these are again provide protective. This protein is protective in nature. So when these proteins are digested, we have studied in vitro digestion. So we have digested milk, these proteins, with the help of pepsin, trypsin, and chymotrypsin, and then those hydrolyzed. We have studied this method, this digestion, in vitro digestion, and not we have not studied this fermentation method or hydrolysis method is going on, so we are studying these. So these studies are done with the help of proteolytic enzymes. Those are pepsin, trypsin, alkylase. There are so many array of enzymes are present, so we can use ultimate aim is to produce bioactive peptides. So peptides, after production of peptides, we have to study whether these peptides have antimicrobial activities or antioxidant activities, or they have got some immunomodulatory activities or opioid activities, or these peptides have some mineral binding activities, or some they are important in heart related diseases. So we have, usually after production of peptides, we have to study all those parameters. So then we can say that these peptides produced in vitro have some health related benefits. So we have studied this in vitro digestion, but we have done that these casein and protein fractions were simulated as like gastric and intestinal digestion with the help of these enzymes. These are enzymes used for in vitro digestion, and we have then compared this undigested and digested casein and whey protein fractions, and ultimately earlier I was telling that we have to study, and to prove that they have got some therapeutic value or not, we have to study these activities. So these activities we have studied with the help of this first is protein profiles, because this is the first and foremost method to study if there is any changes in undigested and digested protein fractions. So this is the proven method to compare the results, that then we can go further. So here we can see that here this is whole milk and this is pasteurized milk, and this milk is from indigenous cattle, and this is from indigenous cattle and this is from Gaddi goat. And we have studied this whole milk, pasteurized milk, then another 70 pasteurized milk at 72 degrees centigrade, and boiled milk. So we have studied these, and if we will go through these profiles, we can see that uh, when it is digested, this milk is undigested, and this is pepsin digested, then this is overnight digestion with this intestine. These enzymes are present in intestine, trypsin, pancreatin, those are used for digestion. And we can see that when these milk is allowed to digest, then these higher molecular protein bands are disappeared, and these specific peptides, low molecular weight peptides, are produced. So here we can see that after digestion, definitely peptides are going to be produced. So we have to identify these peptides. That is the another task we have to fulfill. So here we can say that in pasteurized milk also we can see that these bands are present, but here this array of bands get changed when this milk is digested with pepsin, that is a single enzyme, and when we have digested with trypsin, pancreatin, these enzymes got entirely disappeared. So here we can see that definitely there are some minor peptides present. Those are not visible here, but we have to ultimately test those changes and peptides present, that whether they have got some activity or not. So here in this pasteurized milk, again we can see, and in boiled milk, if we will see that here in pepsin digested means gastric condition, the in gastric conditions, we can see that in boiled milk, these there are another array of proteins, small peptides, those appeared. So here in boiled milk definitely will give a clue that these there may be some other types of proteins, those present after boiling, and these is definitely to be studied. So here again we can see that there are proteins present, and we can compare those almost results. So this is the casein. Again casein, we can see that in casein also these proteins are this is raw milk, and then this is pepsin digested, then this is intestinal digestion. Intestinal digestion is carried out usually in overnight digestion, and enzyme used are trypsin, pancreatin, and here we can see that those higher molecular weight proteins disappeared, and here we can see the same trend again. Here in 72 degrees centigrade pasteurized milk, we can see the same trend. In boiled milk casein also we can see the same thing. And this is the for comparative study we have studied this Gaddi goat casein. Then another fraction was bay, and in bay we can see that those are in even in this gastric condition, these proteins disappeared. So earlier this thermal processing of whey proteins, these proteins were also disappeared, and here when these are digested with the help of in vitro digestion, again these proteins get digested, and we can say that these proteins are not as stable as this casein. So here this Gaddi goat profiles, but here important to note that these proteins in boiled way, these proteins, these are beta lactoglobulin and alpha lactalbumin. These are still remain present in boiled way of Gaddi goat as compared to indigenous cattle. Here we can see that these proteins are disappeared in boiled milk way, but here these remain present. So this is the main comparison, and we can see that these proteins are more resistant to boiling conditions in Gaddi goat as compared to indigenous cattle, those Pahari cattle. Then antioxidant activity. Earlier we have discussed antioxidant activity in raw milk and this casein and protein fractions, but here we have digested, and that is important step that ultimately we have to consume milk, and what is the effect on antioxidant activity, we have to study that in vitro conditions. Earlier I have explained how we have processed the milk that was processed with the help of gastrointestinal enzymes in vitro conditions, and then antioxidant activity was studied. So here we can see that after digestion antioxidant activity has been increased. However, this study will not give we have not got any definite clue, so we have to study again these things. But another method we have tried that another method, FRAP, ferric reducing antioxidant activity, and we can see that when milk was undigested, means as such raw milk, then antioxidant activity was quite low, and when it was in vitro digested, then it has been significantly increased as compared to undigested milk. And this is raw milk, and in boiled milk again when it was simply boiled milk, and here in vitro digested milk, so we can see that definitely there is advantage of digestion, and we can see that after digestion antioxidant activity will get get increased. So then this was we have made comparison between this whole raw milk and whole boiled milk of Gaddi goat milk, and here again we have got similar type of results. Here after overnight digestion, this antioxidant activity has been increased as compared to undigested milk. And here again in boiling conditions, milk was boiled and then digested, overnight digested, again this antioxidant activity has been got doubled. So in case in fraction of this Gaddi goat and bay fraction, again we have got definite trend. So we can see that this antioxidant activity in casein and bay has been increased. So this is again pictorial representations. I have explained this. This again I have explained. Then this antioxidant activity we have studied in undigested and digested. Again we can see that here antioxidant activity, actually this is a qualitative method, and this is another type of verification that but we are getting by quantitative method, we are getting qualitatively or not. So we can see these are yellow spots, those are present in vitro digested milk, and their milk was whether it is pasteurized or boiled milk, we can see that these are antioxidant compounds present in remain present in milk even after boiling. So this was indigenous cattle milk, and again indigenous cattle milk, this is casein protein, and here in casein also we can see that this here here also we can see that these antioxidant activities present, and this is again antioxidant activity in Gaddi goat milk. We can see that these are presence, and here again in bay fraction of Gaddi goat we can see that these spots are present, and even in overnight samples this antioxidant activity has been increased. So that is again corroborating with the effect of quantitative versus qualitative assays. Again, Gaddi goat milk, similar type of observations. So here now we have made efforts to study the we have made this efforts to study those peptides produced after in vitro digestion, whether we can identify those or not. So again, we have gone for this nano LC malditov, and we have purified peptides. Earlier I was telling those were from undigested milk, and that was from Gaddi goat and indigenous cattle. But here, but we have studied in undigested as well as digested milk, and this is important as compared to earlier, because ultimately after when we are going to consume milk, that is ultimately digested, and after digestion those peptides produced that will ultimately affect our system. They those peptides will provide ultimately some beneficial or some negative effect. So we have to see all those things. So here again another gel we have run, and these samples were again subjected to this nano LC malditov, and we have identified around 70 peptides present in milk after digestion, and these are short sequences. These sequences are varying from 4 to 24, and these are definitely have antioxidant activity. Although we have not studied this yet, uh, we have these are our future program. We have to go for that. But the number of peptide produced here, definitely they are showing that if peptides are produced, they have got some bioactive potential. So these are sequences we have identified from different samples. So these are showing samples, and these are sequences. Here again sequences, we have got these sequences are different from each other, and each line has different sequences, and definitely the effect of enzyme on in vitro digestion is observed here, and definitely we can see that peptides produced are of different natures, and we have to see that sequences are also not matching to each other. So here again another uh, these are another lane, these proteins, and we can see that here we have again get MALDI sequences. These are peptide sequences. Again we have got some sequences, MALDI sequences, and then important activities, antimicrobial activities. Then this we have to see this antimicrobial activities. So uh, antimicrobial activity we have studied in vitro digested indigenous cattle and Gaddi goat milk fraction. Actually, again, earlier I was telling about antioxidant activity. That is again important activity in reference to health. Uh, for health perspectives, and antimicrobial activity, again it is important parameter. So we have studied that antimicrobial activity is present in this indigenous Pahari cattle and Gaddi goat milk or not. So we have used some specific strain of bacteria, and those are we have used 5. These are Bacillus cereus, E Coli, Rhodococcus, Shigella, Staphylococcus, and this is the characteristic of culture, and these are the culture details. So we have tested these these microorganism, again antimicrobial activity, those in vitro digested uh, um, milk and its protein fractions, those are produced from indigenous cattle and Gaddi goat milk. So here we have first done antimicrobial activity in digested whey fraction of indigenous cattle milk against Shigella flexneri. So here we can see that minor, we can see that these inhibition zone, if the inhibition zone is wider, or if dia of inhibition zone is higher, then we can say that it has got a higher antimicrobial activity. But if we will see here control, that this is control antibiotic, we have used streptomycin. So here we can see that streptomycin inhibition zone is very higher as compared to our test samples. So definitely there is a very low this antimicrobial activity we can see in milk whey and pasteurized milk whey, and this boiled milk whey and pasteurized milk whey, all those these different conditions we have used milk, and we can see that antimicrobial activity is there against Shigella flexneri, and that fragments we have obtained from indigenous cattle milk. So we can say that this antimicrobial activity is there, but it is not as good as compared to our control. And in this positive control, not control, as positive control. So then we have tested for another strain, that is Shigella flexneri. Again we can see that here we can see innovation zone is there, but again it is not as good as antibiotic, our positive control. So then we have this is again comparison. We can see that here we have made comparison in bay fraction and cage inflection. Earlier I was showing whole milk also, but here this zone of innovation in that overnight digested pasteurized milk, that was pasteurized at 72 degrees centigrade, and that zone of inhibition was 16 mm. So here we can say that in positive control it is 25, but in this our test samples it is 16. Fairly we can say that there it can be used as a used against Shigella flexneri, but again effect is not as expected. So then we have used another strain, that is E. coli, and again we have used E. coli, but here we can see that this is undigested, then digested 3 hours, and all those things I have depicted here. But here important thing is we can see inhibition zone here, that is as good as our antibiotic control here. Here we can see that this is a positive control zone of inhibition, and milk pasteurized at 72 degrees centigrade, that is whey milk. We can see that in overnight digested this whey fraction we are getting this zone of inhibition that is almost or nearly equal to this our positive control. So we can see that definitely this milk this milk whey, pasteurized milk whey fraction that is overnight digested definitely have this antimicrobial activity, and it can be used in combination with antibiotic or alone to treat this E. coli. However, these studies are our initially preliminary studies, and we have to go further to to establish these facts. However, here in this initial studies, we can see that these are at par to each other. Then here we have studied cage infection against E. coli. Here zone of inhibition is there, but again it is not at par with the very less as compared to standard. So this is comparison. We can see that here this positive control has zone of inhibition that is is 27 mm, and pasteurized milk whey fraction at 72 degrees centigrade is 27 mm. So these are comparable, and definitely this whey fraction that is pasteurized at 72 degrees centigrade can be used against E. coli either in a combination with antibiotic or alone. So that is advantage of this study, and we have got this new observation. Then another strain we have used Bacillus. Here we can see that we have not got any inhibition or anything. Here we can see that this is positive control, and but we have not got anything about in these test samples. So here we have got minor inhibition, but again those are not sharp, but in positive control we can see that inhibition is there. Then comparison we can see here These are the zone of inhibition we have measured. Then another microorganism we have used Rhodococcus equi. And here we can see that here we have got this zone of inhibition in indigenous cattle milk, that is pasteurized bay, and here we can see that zone of inhibition is there, but it is not as good as the diameter of this this zone is not higher than this antibiotic control or equal. So here another Rhodococcus equi in cage infection, we can see that here these are inhibition is there, inhibitory zones are present, but again those are not much as compared to antibiotic. So although the purpose of this study, we can see that directly or indirectly we can supplement this 72 degrees centigrade this pasteurized milk casein or boiled milk casein. Those are affected with these type of diseases. We can supplement this casein protein fractions. Those are pasteurized or boiled. So this is comparison. We can see that here in pasteurized milk at 72 degrees centigrade, that is overnight digested, we can see the zone of inhibition is 18, but in positive control it is 22. So then another microorganism we have used Staphylococcus aureus, and here we can see that here we have not got any inhibition in test samples. Only in positive control we can see that large zone of inhibition is there. Then cage infection we have used again against Staphylococcus aureus, but again we have not observed any type of zone of inhibition. So these are again means in positive control we can see the inhibition, but other test samples are not showing an effect on Staphylococcus aureus. So here comparison, we have not got anything, only positive control is showing results. Then earlier I was telling that in this antimicrobial activity of indigenous cattle milk, now I am switching to Gaddi goat milk. So we have started this Gaddi goat milk study. Those are digested casein protein fractions, and that is studied against Staphylococcus aureus. So here in Staphylococcus aureus, we can see that in boiled milk casein that is obtained from pepsin digested, that is gastric conditions, we have observed that gastric hydrolysate of Gaddi goat boiled milk casein showed the largest zone of inhibition, that is 21 mm against Staphylococcus aureus. So here earlier I was telling about this indigenous cattle milk. Now I am showing this Gaddi goat milk. So we can see that inhibition zone, and that is clear as good as this antibiotic control. So I have not gone through all the detail about this Gaddi goat. This is the best results we have observed. So I have shown this only. So now I am switching to anti-hypertensive activities. So this anti-hypertensive activity is involved. These enzymes are involved. Those enzymes are angiotensin converting enzyme. Those play a key role in blood pressure control by producing the vasoconstrictor angiotensin 2. And significantly, goal of hypertension prevention is to inhibit ACE with natural inhibitors to avoid the side effects of synthetic medicines. So now you might be knowing that for control of this hypertension, doctor prescribing captopril, enalapril, fosinopril, ramipril, etc. But those are AC inhibitors and those are synthetic inhibitors. And although those control those blood pressure and other condition related to this heart, but again problem is these are synthetic inhibitors and have side effects, and those are side effects are coughing, taste disturbance, skin rashes, and so many more. And there is challenge today that for this scientists as well as health sectors, that we have to identify natural AC inhibitors. Those can provide this some relief to patients suffering from this hypertension. So we have just studied that is there antihypertensive activities present in this milk or not. So we have studied here again in brief. I am going to tell that we have observed higher antihypertensive activity in overnight digested raw Gaddi goat milk, casein, and whey in comparison to indigenous cattle milk. So we can say that in Gaddi goat milk, antihypertensive activity is present in raw milk. But when milk is boiled, then antihypertensive that is completely lost. So boiling again means it is a negative effect on antihypertensive activity. So here antihypertensive activity, just I have shown that in undigested it is in indigenous cattle it is very low, and in raw milk of this Gaddi goat is slightly higher, that is around 20%, and here in 15%, and in boiled milk casein that is from indigenous cattle, we can see that overnight digest of boiled milk casein of indigenous cattle has around 71%, around 70% anti-hypertensive activity. In Gaddi goat also we can see that this um, anti-hypertensive activities there, but in boiled milk we can see that this anti-hypertensive activity has been completely lost. So this is the disadvantage that whey, when we have to consume whey from Gaddi goat milk, then again this anti we cannot get the benefits of this activity. And then this again we have seen this anti-hypertensive activity in whole milk, casein, and whey. So this is now I am switching to conclusion that but we overall study we have done. This is a part of project, and we have observed that fat and lactose, those are highest fat and lowest lactose was observed in Gaddi goat milk. And earlier just I was telling that casein milk fraction so higher antioxidant activity in comparison to whey milk fraction, and this is observed in all types of milk. And moreover, vitamin C that contents were lower in Gaddi goat milk. That is again another thing. And then we have observed that milk of from all types, that is rich source of saturated fatty acids. However, we have also observed that low level of polyunsaturated fatty acids was present, and that was present only in indigenous cattle milk. Then boiling of milk produced allylic acid, that is a trans fatty acid, and that is present in highest amount in boiled milk of Jersey crossbred cattle, and lowest amount was present in indigenous cattle milk. So again we can see that boiling may lead to production of this trans fatty acid, and those are harmful. And then these are antioxidant activity, these are increased in after boiling in indigenous and crossbred cattle milk. And then we have observed that antimicrobial activity is present in this indigenous cattle milk against E. coli, and Gaddi goat has also shown this antimicrobial activity. And then important again observation was that boiling of Gaddi goat milk may result in complete loss of anti-hypertensive activity. So now thank you much. I conclude my this lecture now. Thank you very much.
First of all, I would like to congratulate you, sir, for your extensive research work. And thank you so much, sir, for your very elaborate lecture on the bioactive potential of native cattle and less explored Himalayan Gaddi goat's milk. I hope many of our participants for many of our participants, this would have been a new area, but really enrich your knowledge with such an informative presentation. Thank you so much, sir. Once again, since there
Thank
are
you.
no queries, I would like to conclude, sir. Thank you so much, sir.
Okay, thank you. So again, I would like to thank the organizer as well as coordinators for giving me opportunity to deliver lecture on this aspect. Thank you very much.
Thank you, sir.